My name is Christopher Sharma, and this is a PowerPoint on nematode and ambrosia beetle interactions with fungi. So when looking at nematodes, we have to consider where they found first of all, because this is on a microscopic scale. So they're found in almost all aquatic and terrestrial environments, and their only stipulations are their availability of their prey and the soil moisture. And fungi are nearly present as much as the nematodes are, but not quite as much. And they're also found in both terrestrial and aquatic environments. When looking at the nematode-fungi interaction, we have to consider its importance to us and why it's a crucial part of the world around us. Nematodes affect both plants and animals with their feeding stylet, so think needle nose pliers. The nematode pierces the skin of its prey to feed on it. This could easily affect the prey from the wound that the nematode leaves, or if the nematode is infected by transferring microbes from prey to prey. The fungi keep the nematode population in check, and the mass created by the fungi after consuming the nematode feeds organisms higher up the food chain. This interaction promotes cycling of nutrients through the food chain. The nematodes and fungi share a synergistic relationship. This means they cooperate together to produce a combined effect greater than the sum of their separate effects. In this case, the food mass that's produced for the organisms higher up the food chain is the combined effect of the two. Even fungi that are known to be non-parasitic become so on roots infected with nematodes to fulfill this synergistic relationship. When looking at this relationship, we also have to consider how prevalent it is. There are around 700 species of taxonomically diverse fungi that are able to attack nematodes. So this is not a niche way of living for 3 or 4 species. This is a very common occurrence in the nematode and fungi microscopic world. When looking at the fungi, we also have to consider that of the 700 species that are present, they don't all trap the same way. They use a variety of different ways. So we have fungal ring users, which use specialized trapping structures differentiated from hyphae. And then we have adhesive hyphae users. We have nematoxin users, which immobilize nematodes before invasion. We have endoparasitic fungi that use their spores to trap. And we have opportunistic fungi, which invade or colonize nematode eggs, their females, or cysts with their hyphal tips. When looking at these fungal rings, the thing that stuck out the most to me was how tightly they hold on to the nematode. Also, personally, I get caught up in looking at the microscopic slides and don't realize that this is a 3D world where there are hundreds of traps waiting for these nematodes, and it's not just a few in a line. So these traps are very much present throughout the nematodes world. While we outlined the 5 varieties of trapping types, these are the specific types of fungal rings that the fungi use. There are constricting rings, there's 5 types of adhesive traps, there's sessile adhesive knobs, stalked adhesive knobs, adhesive nets, adhesive columns, and non-constricting rings. So the main takeaway from this is that most everything is adhesive and it is very, very sticky. And once the nematodes are trapped in the rings, it's very difficult for them to escape. As we can see, the names for these traps define the structures very accurately. In this slide, we see how entrapment occurs. So picture A is just the saprophytic growth with hyphae and the knob, and this is simply the growth of the trap. And then B, it's a newly captured nematode. This is right after the capture of the nematode. Picture C, we see the knobs sticking to the immobilized nematode, and this is about 1 hour after the capture occurs. And D is just the breakdown of the nematode that the fungi is doing. And E is the emergence of new hyphae from the nematode, and F is the trap formation after the degrading of the nematode. The whole infection process from adhesion to new trap formation after degrading the nematode lasts about 36 hours. This slide is simply the last slide. I just blew up the pictures to where you can see it a little bit better than the last one. This is just more pictures of the traps that are occurring. And I really just put these in here because I really like to show the 3D aspect of these traps, and they're not just in a 2D world. And it just shows the amount of traps that are surrounding these nematodes to begin with. The first video shows the swelling of the rings around the nematode, and in this video they've been stimulated the rings have been stimulated in a lab. The second video really shows how much fight the nematode has once it's been trapped, and when they're trapped they're very much aware of it and they're trying to get out actively. And the third video also shows the fight of the nematode, and the narrator speculates that the fungi plan where they set their traps because you can see another one systematically placed close to the captured nematode. The next question we have to ask ourselves is why nematodes? Why do these fungi feature predation on nematodes as opposed to a nitrogen-based diet? Growing evidence is forming that suggests the predatory lifestyle of these fungi is an evolutionary feature. Cellulolytic and lignolytic fungi have changed their diet due to a nitrogen-limited habitat. Based on the fossil record, this evolution occurred in the Permian and Triassic eras as a result of mass extinctions. So this has been going on a long time. Studies also show that nematophagous fungi detect and respond to ascaricides, and ascaricides are the small molecules secreted by many species of soil-dwelling nematodes. So basically, their slime as they are moving along that they leave behind is what the fungi detect. Another interesting topic that's come up recently is the chemical warfare on nematodes in plant production. So in recent decades, the demand for development of biological control agents have greatly increased in plant production. And think about corn and cotton mostly. The interest in nematophagous fungi is due mostly to their potential as a biocontrol agent against plant-animal parasitic nematodes. So, for a substantial number of mycoinsecticides have been developed worldwide. So, basically, people are developing chemical agents of the fungi to protect farmers' corn crops and cotton crops. So now we move on to ambrosia beetle and fungi interactions. The beetle-fungi interaction is as follows. The ambrosia beetles are a group of unrelated scaleton weevil clades defined by a shared ecological strategy, and that's fungus farming. Most bark and ambrosia beetles are associated via fungi. The relationship varies from associations with phytopathogens through enrichment of herbivorous diet with fungal mycelia to strict mycophagy. Instead of eating tree tissues as their ancestors, these beetles carry around symbiotic fungi, they inoculate them into the trees, and they colonize, and are dependent on the fungi as their main source of food, while giving the fungi a place to grow and live. So this slide is more of the beetle-fungi interaction, just more in-depth. Adults and larvae bore into twigs, branches, or small trunks of host plants, so think anything from fig trees to gigantic pine trees. They then excavate a system of tunnels in the wood and introduce a symbiotic ambrosial fungus. So this fungus then provides food for the beetle, and the fungus damages and clogs the xylem, ultimately killing all or part of the plant. The ambrosia beetle and the ambrosial fungus provide food for the beetle, while the beetle provides a safe environment for the fungus. They accomplish this by the beetle boring holes into a plant and secreting the fungus. This burrow in the plant gives the fungus a perfect environment to live. While the fungus lives peacefully, it feeds the beetle. However, the boring and the fungus end up killing the plant, which normally would cut off the food supply for the fungus, causing it to die. Instead of letting this happen, the beetle picks up the fungus and moves it to another spot. Literally, it carries it on with them to another tree, and this gives the two organisms a mutualistic relationship. The range of these beetles is pretty vast. Deploying mycelia of symbiotic fungi as a means of extracting nutrients from trees freed ambrosia beetles from facing the defenses of those trees. And as a result, the amount of host trees that the beetles could use is basically unlimited. So this slide just expands more on the geographic range of the beetles, and the expanded range is because of the ability for them to be able to use many different host trees. And you combine that with their dispersal by flight that allows some ambrosia beetles to colonize enormous geographic ranges. This can be seen two ways. Some species of ambrosia beetle belong to the most widespread insect species on Earth. For example, the Xyloborus affinis, Xyloborus perfornus, and Xylosandrus compactus can be found in all regions with tropical vegetation, including islands as remote as New Caledonia or Hawaii. This capacity also makes ambrosia beetles some of the most invasive insects on the planet. This slide just shows a study that was performed in Papua New Guinea where there were 3,200 described species of ambrosia beetle found, and that's more than any other fungus-growing insect group. However, some ambrosia beetles are still diversifying unusually fast. The beetles are also a dominant component of the beetle fauna of tropical forests, like the ones found in Papua New Guinea. This last slide is just a couple videos of beetle interaction. And the first video is just a cross-section of the beetles living inside a tree. And it's pretty cool because it shows a couple of different tunnels that they have made. And then the last one is a park ranger talking about the effects of these beetles and how they slowly but surely will kill off the trees, and ways to prevent that and ways to identify that they're actually inside the trees to begin with. So that's the end of my PowerPoint. Thanks for listening.